Welcome back to this course on organic chemistry in biology and drug design. Okay. Yesterday we were discussing the one of the most important function of proteins, and that is the catalysis of different reactions that take place in the living organism. Okay. And now again we start with this slide that just to remind you that proteins have several functions in our body, and like you can have structural component, they can play a structural component like making the muscle, where the triple helical collagen plays the important role. Then you have the enzymes that we are going to talk further, and then you have this transport enzymes, transport proteins, or you have antibodies which gives us immunity, and you have receptors which helps in carrying out the signals from one place to another. Okay. Here the particular receptor is rhodopsin. This is the important protein in our eye, and it has got a cofactor. Yesterday I told what is the cofactor, that apart from the protein, there may be another non-protein part which can play a role in a catalysis and also in signal transduction, where there is a the molecule, carotene like molecule, a conjugated molecule, which upon light is isomerized, and then from cis to the trans, and that creates a signal. Okay. So, these are the signal transduction molecules. Now, let us go to the go to that slide where we ended last time. That is, I also want to remind one fact which I told you yesterday, that out of the three structures that we can consider while a reaction goes from the substrate to the product, that you have a substrate and then you have a transition state and you have a product. Okay. And its complementarity is most suitable with the transition state, followed by the substrate, and finally the product is weakly bound. So, product is released in order to allow the next molecules to be turned over into the product. Okay. So, we were discussing the different classes of enzymes, and there are six groups of enzymes which are oxidoreductase, then comes the transferase, then hydrolase, then the lyase, then isomerase, and followed by ligase. I said that this chronology has to be maintained, because IUB has given a number, assigned a number when a new enzyme is discovered by somebody, then they, according to their reactivity, they fall on one or two class. So, they will put a number in front. That front number says that out of these 6, which one does it belong to. Okay. Then we went on to this Michaelis-Menten equation, that how enzymes catalyze from a mathematical point of view, whether which equation, rate equation really fits to the enzyme catalysis. It is called very well known equation, that is Michaelis-Menten equation, and there are two important parameters that we ultimately saw in the enzyme catalysis, and that is the this one, which is abbreviated as ultimately as Km, or which is related to Km, and you have another parameter which is in the next slide, that was the Vmax. Okay. So, these are the two important parameters. You can also say, because Km already contains K minus 1, that is the dissociation of the enzyme substrate complex, and K1 is the association rate of association of enzyme and substrate, and K2 is the rate of turnover of the ES complex into the product. So, these are all these are important, but since this can be this is called a new constant, that is Km. So, we say that the important parameters are Km, and because usually k minus 1 or k 1 are much greater than k 2. So, you can in all practicality you can disregard this k 2, and that makes Km equal to k minus 1 by k 1, which is actually the dissociation constant of the enzyme substrate complex. Okay. So, and what was Vmax? You see, Vmax is related to is k 2, that means the rate of formation of the product, that rate constant, into the enzyme concentration that one has initially. Okay. In theory, enzyme remains like that, because after the reaction it is a catalyst. So, catalyst will be there. So, it can be reused later on. Now, this k 2 is also important, as this is related to Vmax. Earlier I said that there are these parameters Km and Vmax, or before that you can say that I have all these important parameters k 1, k 2, k minus 1, and because these are all related to here, Km and Vmax are Vmax is related to k 2, and Km is related to k minus 1 by k 1. So, you see we generally describe the efficiency of an enzyme by these two parameters Km and k 2. These two parameters we generally use, because Vmax, you see, as you increase the enzyme concentration, you will reach higher Vmax values, obviously, because there are more active sites. So, the true constants are true means under certain restrictions k 2 and Km indicate the true efficiency of the enzymes. Now, same k 2 is also sometimes called k cat, because that is the reaction where the product is formed. Okay. And this is also called the turnover number. That means, it also gives an estimate that how many molecules of the product are formed by turning over the ES complex, okay, turning over the ES complex into the product. So, higher the k 2 means more number of molecules per time is converted into the product. So, it is also called turnover number. So, k 2 is equal to k cat, and that is also called turnover number. Now, the question is that what really decides the efficiency of an enzyme? Now, if you look at the Michaelis-Menten, that not the equation, but the steps that are involved, that E plus S goes to ES and then complex, and ES complex goes to E plus product. Okay. We are just removing the EP as in intermediate, because that is the very transient species. So, we do not consider that. Okay. Now, suppose if I say that this is the k 2, that is the turnover number. Now, an enzyme will be highly efficient if the turnover number is very high, that means the k 2 is very high. At the same time, now on what parameter k 2 depends? K 2 will depend on the concentration of ES, the steady state concentration of ES at a particular time. Okay. So, basically k 2 is dependent on the concentration of ES. Now, concentration of ES is dependent on these two parameters k 1 and k minus 1. Okay. So, that means, if I have more ES that and more k 2, then it will be then I can say that the efficiency of the enzyme is very good. Okay. That means, to have high efficiency, what we need is high concentration of this ES and a high value of k 2. Okay. Now, this is interesting, that if some enzyme is there which has got a very high Km value, but its K2 is also very high, K2 is large. That means, it has got a large turnover number, but it has got a high Km also. Now, the question is this enzyme very efficient versus an enzyme where there is a low Km and also a very high turnover number, high K2. Okay. This is also high. Question is, which enzyme will be more efficient? Now, you know that rate, this is Km, low Km means less dissociation of ES, because Km is defined as is roughly the dissociation constant of the ES complex, the enzyme substrate complex. Okay. So, low Km is very good, because that will at a particular time the concentration of ES will be high, because the dissociation will be less. High Km on the other hand means that the concentration of ES at a particular time is low, because the dissociation is favored, because it has got a high Km. Okay. Now, that means here I can say that the rate of the reaction is actually proportional to, inversely proportional to Km. Lower the Km, you get higher concentration of ES. Okay. And at the same time, rate is also, now rate is directly proportional to K2. Larger the K2, the higher is the turnover number of the enzyme. So, if you combine this, you ultimately you will see that rate will be dependent on K2 by Km. In the books, you will see that it is equal to K cat by Km. This ratio actually decides the efficiency of an enzyme. I have done in a just variation method, but you can ultimately prove it by doing all this, take the Michaelis-Menten equation, and then you can derive this aspect that the efficiency of an enzyme is dependent upon K cat by Km. But without going into deep mathematics, it can be very easily said, because rate is inversely proportional to Km, rate is directly proportional to K2. So, an ideal enzyme, if K2 by Km is very high, that is a very efficient enzyme. Okay. Now, this Km, again I remind you, Km has another way of defining, that is the concentration needed to reach the half of Vmax, the maximum value what we can get, that is called the Vmax. Half of Vmax, the concentration needed to get half of Vmax is called the Km, right. So, I remind, because it is a concentration, so Km has a unit of concentration. Okay. It is a concentration unit. It is also the ratio of two rate constants. But remember, K minus 1 is a unimolecular reaction. When ES goes to E plus S, it is unimolecular, it is a first order type reaction. So, here the rate will be, here the constant has a unit of concentration per time, but K minus 1 has two concentration terms. So, per time. So, ultimately the one concentration term will be crossed out, and the time also will be crossed out. So, ultimately this gives a concentration unit. Okay. So, in both ways you get the same thing. That should happen also, because both are pointing to the same parameter. Okay. Now, then we also introduce this double reciprocal plot, that how can we get the values of Km and values of Vmax. That is done by doing a double reciprocal plot, a Lineweaver-Burke plot. You know that this is done by taking the reciprocal of the equation that you get directly from Michaelis-Menten equation. So, you have to do a reciprocal of that equation, and you get a straight line. So, 1 by V 0 versus 1 by S will give a straight line like this. Okay. Remember, this V 0 I told you, it is the velocity at the initial point, because the enzyme velocities ultimately the maximum, it will be maximum in the beginning when the enzyme is available in total quantity, but as soon as some active sites are being filled up, the rate will slowly decrease. So, the initial rate, this Michaelis-Menten equation, this is V 0, that means this is the initial rate. At the initial rate, the line remains straight line. So, if you take very quick data points in the very beginning, say 1 minute, 2 minutes, 3 minutes. So, you get that straight line curve and then you extrapolate and you get the value of the V max, okay, value of the V 0, the initial velocity. And so, these are the things, this is the intercept minus 1 by K m and this is 1 by V max. Then we went to the factors, these are all done. I think this is the slide where we ended last time. That is the mechanism of, we want to know the real mechanism, that means from molecular aspect what really happens during enzyme catalysis. And for our purpose we have chosen the protease class of enzymes. I told you there are 4 different classes of proteases. One is called serine, one class serine protease, where serine is the nucleophilic nucleophile that assists the hydrolysis of the peptide bond. Then there is this cysteine proteases, cysteine where SH is the nucleophile. Cysteine proteases are also called thiol proteases, and then you have a metalloprotease where a metal ion is required, and that means that is a cofactor. And then another one is called aspartyl protease, where aspartic acid remaining at the active site, they assist in hydrolyzing the amide bond or the peptide bond, the same thing. Now, let us talk about, so we have, this is this example of serine protease. One enzyme that does that, you already know that, that is chymotrypsin. Chymotrypsin recognizes aromatic amino acids and cleave the peptide bond from the carboxy end, okay, from the carboxy side. Now, I told you that serine is the nucleophile here. So, mechanistically what is happening is that CONH, and you have this protein chain on this side, and on this side you have the R, that is the aliphatic side chain, and you have the other, you have now NH again and then CO. So, the other part of the protein. Now, what we are saying that there is a serine amino acid that comes and attacks this goes here, that comes back, and the amide bond is lost. So, this is the mechanism, that means, but this is not the complete mechanism yet. What you have done is you have broken the carbonyl carbon nitrogen bond, but so one of the peptide that is going out from the right side, that means from the C terminal side, that has got the NH2 free amine, but this one where the which is attacking the carbonyl, that ends up as an acyl, what is called an acyl enzyme complex. That means O it becomes actually double bond O when the lone pair comes back again, and you get this enzyme is now tied up with this. Okay. So, this is the acyl enzyme complex. So, I think it is clear what I am saying, that this is our nucleophile that attacks the carbonyl, breaks the carbon nitrogen bond. So, this protein is now free with an N terminus and C terminus, but this protein on the left side, that has got the C terminus attached to the enzyme serine, and this is what is called acyl enzyme complex, because this is an acyl group. Okay. Acyl enzyme complex. Now, water comes, the next step is water that should come, and again by the same type of mechanism the serine is released. Okay. The serine which gets its proton back, what it has already donated earlier. So, that was the mechanism. However, there is a serious flaw in this, in the sense that this serine hydroxy is just an alcoholic hydroxy group. This alcoholic hydroxy group, alcoholic OH cannot hydrolyze a peptide bond because OH is not sufficiently nucleophilic to attack the carbonyl. So, what happens here? People then realized when they checked many of these serine protease class of enzymes, they found that in the enzyme there are always 3 particular amino acids which are present at particular sites. There is a serine which is present in for chymotrypsin, there is a serine which is at amino acid 195. That means 195th amino acid is a serine. Okay. Then you have a histidine. Histidine is basically, remember histidine is your, it contains imidazole. So, N, then another N, and this is the structure, sorry, not this double bond, this is a double bond here and a double bond there. So, maybe I can erase that extra double bond. Yes. So, this is the histidine, it has got a NH. Okay. So, a histidine is always there at position 95, and then there is an aspartate which is at 102. Now, these numbers means the position, this number means the position of the amino acid. Okay. The 195th is serine, 95 is histidine, and there is an aspartate at 102. So, it cannot be a continuous chain because you see 95, 195 and 102, 95 is in between these two. So, the possibly it will be like this, that first your 95, then 102 that comes. So, it may be like this, that you have a 95 at some position, then you have a 102 somewhere here, and then that turns back, and then you have 195 here, because that is the only way you can have that. So, this will be your 95, then the 95 goes to 102, and you actually go from this N terminus to this C terminus. So, 195th is a serine, 95 is histidine, and 102 is your aspartic acid, aspartate. We generally call aspartate or glutamate in biology because at the biological pH they will all the time they will be ionized. So, that is why remember glutamate is different from glutamic acid. Glutamate is the ionized form of glutamic acid. Okay. So, here it is aspartate. Now, what happens here? People have found, once they notice that these 3 amino acids are present in the serine class of proteases. So, that means these 3 are important in doing the catalysis. And finally, this is the mechanism that is suggested, that you have the, let us see where it has started. It has started from, so this is serine, in this case it is from the left side. So, this is your aspartic acid which is present as aspartate, CO2 minus. Okay. Then you have the histidine, this is the histidine, and finally, you have the serine. So, what is activating serine? We know that an alkoxide is a very good powerful nucleophile and not the alcohol. Now, the making of this alkoxide kind of thing is assisted from these other amino acids, aspartic acid, then histidine. So, what happens here? Aspartate negative charge that abstracts the hydrogen of the histidine, and this now flows here in this direction, and sorry, not that direction, I am sorry, it flows in this direction, and then the nitrogen becomes highly nucleophilic or highly basic. It takes up the hydrogen and releases, and then takes up the hydrogen. So, basically you are making an alkoxide now, and that attacks the carbonyl. That is more powerful, much more powerful than the alcohol itself. So, if you suppose remove the histidine by some process in the enzyme, in chymotrypsin, you do some engineering to remove the histidine, or to, then you will see that there is no catalysis. or even if there is catalysis, that will be very less. So, almost the catalytic activity is gone if you remove this. Catalytic activity will be gone 100% if you remove serine, because serine's presence is absolutely necessary. Catalytic activity is also gone if you remove the other two also, like histidine or aspartate, and that was the beginning, that when they removed aspartate, they found that the activity is decreasing. Then they removed the histidine and they found that the activity is almost gone, and when they removed the serine, they found that the activity is lost completely. So, serine is absolute necessary, but along with that histidine and aspartate, they are also required. So, now you see a nice relay process. The carboxylate takes the hydrogen, in turn activates the histidine, and histidine in turn is activated as a base, and it now takes the hydrogen out from the serine. So, making virtually an alkoxide, and that is now able to attack the carbonyl. Okay. So, it attacks the carbonyl because it is a powerful nucleophile, and then so what intermediate you will get? You will get an intermediate like this. What is let us see which one. Here is the histidine. Actually, I can write it. It better that I should write it, then it will be more clear. So, what we are saying, that there is a serine, there is a histidine, and then there is an aspartate. Okay. So, we are writing COO minus, and here is your peptide bond. Okay. Remember, for if it is chymotrypsin, then you have this R as some aromatic group. So, the mechanism suggested is a charge, this is called a charge relay process. This takes the hydrogen, that goes here, that comes there, makes the alkoxide, and that goes, and initially that will form a what is called a tetrahedral intermediate. Initially that will not be broken, the negative charge goes to the carbonyl oxygen. Okay. So, O, then CO, then O minus, and this is attached to the left side of the peptide, and this is the NH, and this is the other side. And so, this is, so you are developing a negative charge in the, this is in the intermediate, this is not the transition state, because this is the intermediate where the bonds have been formed. In transition state, bonds are formed partially. Here the bonds are formed in fully, but this is not a very stable system, because you have a negative charge on oxygen. Now, enzyme is also extremely clever, because you know that if you can stabilize the intermediate, that means the reaction will be faster. Okay. I told you the intermediate is, what is intermediate? That if in a reaction, it is a two-step process, serine protease, because the first reaction is this. So, you get an intermediate via a transition state, where that will be delta negative, but now we have an intermediate. Intermediate is, there is a postulate which is called Hammond's postulate, that we do not know exact structure of transition state, because these are they are extremely transient species, but what will be its structure? We can approximate the structure by saying that the structure which we can confidently tell, whose energy is closer to the transition state, so its structure will resemble that. That means, if this is the intermediate, then transition state will be structurally close to the intermediate and not the substrate and not the product also, because this is energetically much closer to this. So, this is intermediate. Now, the enzyme stabilizes the intermediate by having what is called an oxyanion hole, which is, so there is a groove here which is positively charged, or which has got ability to donate hydrogen, and there can be a stability by hydrogen bond. Okay. So, this oxyanion, the intermediate is an oxide, and there is what is called an oxyanion hole. So, that actually stabilizes the O minus. Okay. So, what is the next step? Next step is this comes back and the NH leaves. But this is not the complete mechanism I told. This is one of the protein part is gone. The other protein part is now attached to the protease via the acyl enzyme complex. But the next step is easy, water comes and then breaks the acyl enzyme complex, hydrolyzes the acyl enzyme complex. That is what was shown here, that you will see all these charge relay processes. The other important point is that also I should tell you that when the acyl enzyme complex is hydrolyzed by water. So, this is your serine, you have the histidine. Now, the structure of the histidine, what we drew earlier was NH on this side and N on that side, but now this has got the H, and this nitrogen is devoid of the hydrogen. There is a transfer, this hydrogen belong to the serine, and the hydrogen belonging to this nitrogen is now taken by the aspartic acid. So, aspartic acid is momentarily converted into aspartate is converted to aspartic acid. So, now what happens, water comes, it is a reverse of the charge relay process. Earlier what happened, exactly the reverse step. Reverse step means water comes, breaks the, so first it goes there, it comes back, and then this regains its hydrogen, what was momentarily with the imidazole moiety of histidine, this comes back, and this nitrogen gets its own hydrogen from the aspartic acid, making the aspartate. So, you see, first electron flowed to the serine, and the second series of steps is that the hydrogen is, there is again a relay process, but here it is a proton transfer. So, the proton is transferred to the serine, and the imidazole gets back its proton, which was momentarily with the aspartate. So, the enzyme is regenerated and it can do the catalysis again. In similar mechanism happens in cysteine proteases also. Here the SH is activated by again an imidazole, but sometimes what was found that in some thiol proteases or cysteine proteases, there could be a catalytic, by the way, this is called a catalytic triad, okay, because there are 3 amino acids that are present, catalytic triad. In some of the thiol proteases need this aspartate and as well as the histidine, but because sulfur is possibly more nucleophilic. So, you may not need the aspartate, maybe imidazole itself can be able to pull out the hydrogen. So, in this case thiol protease, you have the dyad mechanism as well as the triad mechanism. Okay. Triad means now you have the same aspartate, histidine and cysteine, or you can have only histidine and cysteine. The one of the very well known thiol protease is what is present in papaya, and you know, papaya juice is usually given while you cook meat, because that can soften the meat by hydrolyzing some of these some of the peptide bonds that are present in meat. Okay. Now, this is the what is the called metalloproteases. I told you in that case a metal ion is what is activates the carbonyl. So, there are, this is a metal ion mechanism where there is an there are 2 types of actually metalloproteases. In one case the metal activates the carbonyl, and this is a glutamate moiety that attacks the carbon, and this is stabilized by zinc. So, as if the zinc is providing the oxyanion hole that was earlier present in chymotrypsin. Here it is the oxide, that carbonyl becoming the oxyanion, that anion is stabilized by zinc. Okay. And then it comes back and the NH goes out. Okay. So, that is the it goes out and it is momentarily attached to the aspartate, in this case glutamate, and then water comes and hydrolyzes this intermediate. Okay. I am not going into the details, the structures are all here. So, you can see one by one from here. Other mechanism is that zinc and a carboxylate, a glutamate, can activate the water molecule. Like serine was activated by histidine and aspartate, you can activate a water molecule by what you can do, the aspartate can take the hydrogen from water and the oxygen was chelated to zinc. If you chelate the oxygen to zinc, then the acidity of this hydrogen increases. So, the aspartate is able to abstract the hydrogen from water, and this virtually becomes hydroxide, and the hydroxide being a strong nucleophile, that can come and attack the carbonyl and hydrolyzes the peptide bond. So, that is the mechanism of metalloproteases. One of the, there are two mechanisms I have shown. In one case, the glutamate directly attacks and the carbonyl is stabilized by zinc. In the other case, water, no glutamate attack to the carbonyl, it is the water directly attacks the carbonyl, but here zinc and glutamate, they actually mutually cooperate to activate the water to make hydroxide. Okay. And the last one is what is called aspartyl protease, where there are two, in the active site there are two aspartate residues. As one residue is present as the acid form and the other is the carboxylate form, and you have water. So, what happens here now, again there is a relay donation of charges in a relay mode. So, the oxygen, the oxide of the carboxylate takes up the hydrogen from water, this becomes hydroxide and this becomes O minus, that is actually that was stabilized by this carboxylic acid, which is in the acid form. Okay. So, the next step is, this takes up the next step is this hydrogen, actually it is not given here. The next step is, after the hydroxide is attacking here, the peptide bond breaks. Only the purpose of having two aspartate is, one aspartate is helping the release of hydrogen ion from water, so make hydroxide. The other aspartate in turn is giving stability to the oxyanion that is being generated, like what I have shown here, and that completes this 4 classes of proteases. Okay. Only the protease, not any other enzyme. Okay. In the next class we will talk about, next session we will talk about the how to control the activity of an enzyme. Okay. Thank you.